That's not waste. Yes, now it's recording. Okay. State the function of the human teeth. Quickly, this is too easy to even waste any time.
Mechanical digestion.
Yes, mechanical digestion. This is enough if the question is out of 1 mark. I want you to understand the nature of the questions. But because it says state, you will say mechanical digestion, and this is enough. If they said explain the function of the human teeth, what will you do? And it's out of 2 marks. How will you answer this question? Will you just say mechanical digestion and stop there?
It breaks large molecules of food into small molecules, small pieces, and it
Okay.
chew and grind food to increase the surface area.
Very good. This is the difference between the command terms state and explain. State means just say a word or a sentence. Okay. So state the function of the human teeth. It is enough to answer by mechanical digestion. If they say describe or explain, then you will have to elaborate a little bit more. You will say mechanical digestion and you will mention the definition, which is the breaking down of molecules, making down of large molecules into smaller molecules. That's it. You will not say large insoluble into smaller soluble, because this is not the definition of mechanical digestion. This is the definition of what?
Chemical digestion.
Exactly. And you will elaborate, you will write everything you know about mechanical digestion, and you can even give an example. So if this was explained out of 3 marks, you will say mechanical digestion, that is 1 mark. And the second mark, because we are breaking large molecules into smaller molecules, that's the second mark. And we do that to increase the surface area, just like your friend said, for the enzymes to work next. And this will be the 3rd mark. And you can even write one example. Example, what's the easiest example we use in mechanical digestion? Give me an example. Part of the digestive system that does mechanical digestion. Give me an example. What do you see here? Did I lose connection? Hello? Hello? Can you hear me?
Yes, we can hear you.
Okay, give me an answer. Give me one example for mechanical digestion.
Teeth.
Exactly. Another example.
Churning in the stomach.
Exactly. Very good. A third example.
Boil.
Exactly. Very good. So here, I want you to understand the difference between the command terms. State, you will write 1 word or 1 sentence because it will be out of 1 mark. Explain or describe, it will be out of 3 marks or 2 marks, you will elaborate. This is very important to understand because a lot of you lose marks because you do not give the needed information. You give less information, even though you know it. So look at the command term and look at the number of marks provided on the question. If it is 1 mark, you will write 1 word or 1 sentence. If it is beyond 3 marks, 3 marks or more, you will elaborate. You will write everything you know about that point. This is very important because you all know the information, but you don't write the information down. I don't know what is the reason. State the name of the visible outer layer of the teeth. Again, it states, the command term is state, and the number of marks allocated is 1. So you will just write the names. You don't need to elaborate anything. What are the names of the visible outer layer of the teeth?
Enamel.
Very good. Enamel, that's it. Even though you know more information about enamel, it's made up of calcium, it's the strongest tissue. You don't need to write that because they said state and it's out of 1 mark. Now compare this command term with the explain out of 3 marks. I want to teach you how to write an essay question. Explain, and out of 3 marks, you will write everything you know about this point. Explain the process of tooth decay in humans. Will you write acid? And that's it, because some students do that. And then they ask me, why did I take 1 out of 3? Because you did not write all the information. What will you see here? What is the process of tooth decay? I'm waiting.
food forms plaque, which bac- bacteria then secrete acid that dissolves the enamel.
There is a step missing. You said that the bacteria will produce acid. Good. And this acid will dissolve the enamel. Correct. Now, how will the bacteria produce the acid? What is the process that the bacteria will do so
Aerobic
that
respiration.
exactly? Okay, so you get a 2 out of 3. That's why you finish the answer, you go back and read it in case you miss something. So you will say that there will be food remains in your mouth. You don't brush your teeth. The bacteria inside your mouth will feed or they will do respiration. And to be specific, what type of respiration?
Anaerobic.
Correct. Anaerobic respiration. You know it, you write it down. And they will produce lactic acid. And this lactic acid will dissolve the enamel. Write every piece of information on a separate sentence so that it is nice and neat. And if you want to write the equation for anaerobic respiration, you know it. C6H12O6 will be broken down to give me what? The lactic acid. You know the information, the formula for lactic acid, even though you don't need it here, but you know it. What is the formula for lactic acid? Who will remind me? Read it in English.
C2H5OH.
1 minute. What did you say?
2C2H5OH.
No, this is alcohol. Lactic acid, we said it is it's half the glucose. So it is C3H6O3. If you remember, this happens in the muscles. When we are doing vigorous exercise and our muscle breaks down the glucose in anaerobic respiration and we produce lactic acid. What you said, C2H5O8 is alcohol. This is in fermentation in yeast. This is C2, this is C6H12O6. Glucose is broken down into alcohol and carbon dioxide. Carbon dioxide and alcohol, C2H5OH. This is alcohol. This is fermentation in yeast. This is not what we want. We want this, where the glucose is broken down to 2 lactic acid molecules. And this happens, or we know it, we studied it when we studied anaerobic respiration in our muscle cells. It produces lactic acid, and then this lactic acid is carried by the blood and it goes to the liver where it is oxidized back to carbon dioxide and water. This is not needed here. You don't need to write this equation, but if you write it, you will not lose a mark. You write it usually when we are talking about anaerobic respiration in the muscles. But you will write those 3 points. They have to be there. There will be food remains, and the bacteria inside your mouth will feed by anaerobic respiration, and they will convert this food into lactic acid, and this acid will dissolve in them. Those 3 points will give you 3 out of 3. Here, let's jump to the next question. This is classification. Mammals can be classified according to the position and the shape of their teeth. Figure 1.2 shows the skulls of 7 mammals. Let me zoom down. So you have A, B, C, D until G. Okay, write the letter of each species, A to G, in the correct box in the key. This is very good, so we can practice that. A lot of you get confused with the classification. Here, they will give you some criteria, and you will use it as a puzzle to name those mammals. Okay? So 1A, 2 or more different types of teeth. We'll go to 2. Let's go to the picture. Now, don't rush. In this type of question, you should not rush. You have to do it carefully so that you get the answer right. 2 or more different types of teeth. Let's look at A. Do you have 2 more different types of teeth? And B. Let's look at A. You have 2 more different types of teeth, yes or no? Quickly. Yes or no? A. Can you see 2 different types of teeth in A, or you do not see 2 different types of teeth? I want yes or no. Did I lose connection? Adeya.
Yes.
Okay, answer. Yes. So A is included. B, do you have two different types of teeth or not two different types of teeth? This is the truth, by the way. So B, yes or no?
Yes.
Yes. Okay. C, yes or no? Quickly. And C, you have two different types of teeth or you do not have two different types of teeth? Quickly. What do you see? Hadia.
Yes.
Okay, stay with me. D, yes. You're wasting so much time. E, do you see two different types of teeth or no?
No.
No, so I will not include E. Solve it like this with a pencil. This will really help. F, I don't see any teeth anyway. And G, yes, I do see different types of teeth. So A and B and C and D and G show me two different types of teeth. So two or more different types of teeth, go to two. I'll go to two. They have a wide gap between the front and the back teeth in both jaws. Let's go see who has a wide gap. Yes, A does in both jaws. B does in both jaws. C, no. So I will remove C now. D doesn't have a wide gap between the teeth. E is already not included. F is already not included. G is included. So now we narrow it down into A and B and G. Okay. So wide gap between front and back teeth in both jaws. Go to 4. I'll go to 4. No incisors in upper jaw. Let's go and look for no incisors in the upper jaw. A, no, it does have incisors in the upper jaw. B, it does have incisors in the upper jaw. They said no incisors in the upper jaw. Yes, this has no incisors in the upper jaw. So this has, so it's D. So A, no incisors in the upper jaw. No, this has incisors. So this is not included. I'll exclude that. B, it does have incisors. So this is excluded. It's only G that has no incisors in the upper jaw. So let's go to 4. No incisors in the upper jaw. Its same as cervix, elyphus. This is G. You understand how we did it? Okay, let's go to B. Incisors in both. Now, when you get to one letter, then you will go back to the very beginning of the classification. No incisors in the upper jaw, cervix, elyphus. This is G. So now we will remove G altogether. We will erase everything we did here because we'll go back to the beginning of the classification. And G, you already have its name. So G is no longer within the classification. I already named mammal G. Let's go back to the beginning. Two or more different types of teeth. Go to 2. Two or more different types of teeth. We already did it before. A is included because it has two or more different types of teeth. B is included. 2, C, and D. E, no, it has only one type of teeth. F, it has no teeth altogether. And G is already out. So G is already out. We already named G. We'll go to 2. Let's go to 2. They have a wide gap between the front and the back teeth. Let's go back. There's a wide gap, yes. In A, there's a wide gap, yes. In B, in C, no. I'll exclude C. D, it does not have a wide gap. E and F are already out, and G is out. So now I'm left with A and B. So they have a wide gap between the front and the back teeth. I'll go to 4. Okay, let's follow the orders. I'll go to 4. No incisors in the upper jaw. We already named this mammal, so I'll exclude this. Let's go to B. Incisors in both the upper and the lower jaw. Okay, let's go to the two mammals that we have. Incisors both in the upper and the lower jaw. Yes, that's both A and B. So if the incisors are found in both the upper and the lower jaw, I'll go to 5. Okay, I'll go to 5. 5A. Incisors on the lower jaw are longer than the incisors on the upper jaw. Incisors on the lower jaw are longer than the incisors on the upper jaw. We said we have A and B. So this is A or B. Which one has the incisors in the lower jaw that is longer than the upper jaw? It's very obvious. A or B? Give me an answer and don't stay silent. A or B? Let me choose someone. Layla. Which mammal,
Yes.
A or B, has an incisor in the lower jaw that is longer?
A.
No. The incisors here are the same size. Look at B. Which
Oh,
one has
sorry, أيوة. B. Lower.
used? Exactly. So, incisor, where are we? Incisor on the, yes, here, 5A. Incisors on the lower jaw are longer than the incisor on the upper jaw. Its name is Marcus Rufus. This is B. So we already named B. So I will exclude this altogether. Do you understand how we did it? Because a lot of you don't like this type of question. So B is already named. B is out. I will not include it again. Fine? This question takes time, whether you like it or you don't. Let's go back again, or let's continue. Incisors on the upper and the lower jaw are similar in size. Let's go to the mammal and look at incisors on the upper and the lower jaw that are similar. B is out. So is it A or C or D or E or F? Incisors on the lower jaw are the same size. G is already out. So A or which one? It's very obvious.
A.
It's A. Very good. Okay. So incisors on the upper and the lower jaw are similar in size. It's A. So I'll put A here. So A also will be out with a pencil. It will really help you so you don't get confused. So A is already named. A is out. Let's name the rest. Incisors on the lower jaw project forward. Let's go here and look at incisors projecting forward. So A is out, B is out, C is out, oh, C is in, B is in. And E, which one? C, D, E, F has incisors that project forward. Again, it's obvious.
C.
Yes. So we have incisors that project forward. This is C. Write C here. I exclude this. Good. Incisors on the lower jaw do not project forward. Let's go and look for incisors that do not project forward. It's obvious, I think. So C is also out. D is in. It does not project, doesn't even have incisors in the lower jaw. E, they're all the same teeth. F, tell me, which one has incisors in the lower jaw that does not project forward? No, it is D. Tell me. G is out, C is out, A and B, so you can't choose from A or B or C. You can only choose from D or E or F. Those three. Which one has incisors in the lower jaw that do not project forward?
D.
Yes. So I'll go to D and I'll write this here. So I'm done. Okay. So these types, oh, I still have two here. All teeth of similar shape. This is easy. So again, this is out now. All teeth are of similar shape. This is easy. E or F, these are the only last two.
E.
E, of course. All teeth are of similar shape, so I'll write E here. So again, this is out. No teeth on either jaw. Very easy. This will be the last one here, F. So these type of questions, they're easy, but just take them slowly and step by step. And every time you name a mammal or an animal, cross it out so you don't get confused. Okay, killer whales are mammals. State two internal features you would expect to find in a killer whale that would not be in a fish. Now, this is a nice question. Because here they're not talking about the external features of a mammal, the internal features. Can you think of internal features in a mammal that are not found in a fish?
Lungs.
Correct. Very good. The fish, they have gills. So I will write lungs. What else?
They're warm-blooded.
They are warm-blooded, correct? They are warm-blooded. Let's write all of them. What else? What
Double
is in a fish?
circulation.
Exactly. We studied this in a fish and it is inefficient. Fish, they have a single circulation. Mammals, they have a double circulation. Okay. And they have a sophisticated respiratory system, lungs and a diaphragm, etc., etc. Can you think of something else? They
Uh,
also have,
Mammals
uh,
have two atria and two ventricles.
yes, correct. So they have four chambers in their heart. Correct. Correct. Fish, they have two chambers. Excellent. Also, they have also two sets of teeth, one milky teeth for when they were children, and one permanent teeth. And then the milky teeth or the deciduous teeth falls off, and then they have permanent teeth. Very good. Okay, I thought you would not be able to get this question. State the name of the group of animals that includes mammals and fish. Mammals and fish are grouped together in what? They are both what? They both have a backbone or they don't have a backbone. That's very easy.
Great.
Excuse me, my dear. What did you
They
say?
both
I did
are
not hear.
vertebrates.
Vertebrate.
Vertebrates, correct. Now, I want to surprise you and tell you that the first 2 pages of this exam is year 9. This is all year 9 material. So in case you did not take the core with us last year, please focus on year 9. Those who took year 9 last year, they already know it very well and they can study it lightly. But those who did not take the core last year, please focus on that because I thought they used to get one question or two, but here those 2 pages are all year 9. It's all year 9. Now, 2. Explain why it is important that large insoluble molecules are broken down by chemical digestion. Why is chemical digestion important?
To increase surface area?
No, chemical digestion does not increase surface area. For chemical digestion to start, surface area should have been already increased in mechanical digestion. Why is it
For
important?
easier absorption, sorry.
Exactly. Large insoluble molecules are broken down into small soluble molecules, so they're small. So they become small enough to enter the cells. Again, this is year 9, oh my God. To enter the cells so that they can be absorbed into the cells. And how are they absorbed? What are the methods of absorption?
Osmosis,
Correct. Osmosis if water. What
diffusion,
else? But,
active
but
transport,
water is not active transport, yes. What else?
Assimilation?
Assimilation is entering inside the cells. To make assimilation successful, okay, absorbed into the cells is the same as assimilation, good, but it is taken into the cells by active transport and diffusion. Actually, osmosis is not included here because water is not broken down. Osmosis includes water, and water is not digested. Water is the only nutrient inside the body that is not digested. Okay, no enzymes work on water. We don't digest water. So here it shows, so it is small enough to enter the cells, to be absorbed, to be assimilated, okay? And it is absorbed by active transport or diffusion. And you don't need, of course, to elaborate on active transport and diffusion. This is enough. State the names of the substance that is the solvent for most molecules that have been digested by enzymes. What is the universal solvent inside your body?
water,
Excuse me?
water,
Water. Yes. Again, this is year 9. All this until now, it's year 9. This is scary. Okay, now I have a graph here. Of course, you will read all this. I just want to jump. Here you have pH and you have enzyme activity. So this graph is showing you the effect of pH on enzyme activity. Okay. Use the information in figure 2.1 to support your answer. Describe and explain the roles of the two protease enzymes A and B in the alimentary canal. So describe and explain means write everything you know. You will describe and you will explain. Describe and explain. So you will go to the first graph, the one with the solid line, which is enzyme A. You have a key here. You will describe it. Can you describe enzyme A? Describe and explain. What will you say about enzyme A, graph enzyme A? I want to choose someone. Just give me an answer. Quickly. What do you see?
It works in acidic mediums.
Yes, but you jumped. I want you to describe, you will break down the graph into 3 parts. This is the first part, this is the second part, and this is the third part, like we always do, and give numbers. So you jumped and you told me it works in acidic medium. Correct, but I want more elaboration here. How many marks is this question? 6 marks. So we're not in a hurry. What would you say? Hmm? How will you
Enzyme
describe the
A
graph? Yes.
in pH from 1 to 2.
Yeah.
The enzyme was started working.
What do you see as the pH is increasing from 1 to
As
2,
the
correct?
pH
What
is
does happen?
increasing, the enzyme
Yeah.
activity is rapidly increasing.
Yes, correct. This
At
is the describe.
pH
Very good. Huh?
at pH 2.5,
Yes, correct.
it's the optimum pH for the enzyme to work. The
Excellent. Very
enzyme
good.
activity is at its highest.
Perfect.
From
And then what do
pH
you say here?
2.6 till 5, the enzyme started to denature.
Excellent. What do you say about denaturing?
The shape of the active site is not complementary to the substrate, so there are
Perfect.
no enzyme substrate complex.
Excellent. Excellent. Look at the difference between your answers. At the beginning, you told me it's a enzyme that works in acidic pH. That's it. And look at now what you said. You broke it down into a perfect elaboration. This is described. You told me that from 1 to 2 it will increase, and then at 2.5 this is the optimum, and you used the word optimum, and then it will denature, and you discussed what is denature. This is perfect description. Now, can you explain? This is which enzyme? Why does this enzyme do that? This is which enzyme? Which enzyme do you know that works in these conditions?
Pepsin
Pepsi.
enzyme.
Very good. The pepsin enzyme that works where? In which part of the digestive system?
Stomach.
Stomach.
Excellent. And what does it break down?
Protein into amino
Protein.
acids.
Perfect. This is perfect. Now, we finished enzyme A. Who will give me the describe for enzyme B? Hmm? Now, just what she did, exactly what she did. Quickly, 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 quickly.
I will say that enzyme B is trypsin.
Yes, you correct, but you jumped the explanation. I want the describe.
The optimum pH is at 10,
Yes, I want this part of the graph. Can you explain it, please?
and the enzyme start to denature after start to denature from pH 3.
Okay. It
I
denatures
will
at pHs away from the optimum. You are correct. So it can denature here and denature here. But as the pH is approaching the optimum, you said the optimum is 10. When the pH is close to 10, the enzyme is denaturing or the rate of activity is increasing.
reaches 10.
If it is close to 10, we said 10 is the optimum, correct? Because this is trypsin. It will increase. Okay, very good. This is pH. And none of you mentioned anything about kinetic energy, which is very right. When do we mention kinetic energy? When we are talking about effect of pH on enzyme or effect of temperature?
Effect of temperature.
Very good. So again, both of you gave me a perfect description and a perfect explanation. For enzyme B, we will also say that as the pH is increasing from 3 to 10, the rate of the reaction is increasing because it is approaching the optimum. And you can say as it approaches the optimum, you have a faster rate of reaction, faster enzyme substrate complex, more production of products. Just do not mention anything about kinetic energy. No kinetic energy when it comes to pH. And then 10 is the optimum maximum rate of reaction. And then after 10, it will start to denature. And you don't have to elaborate denature 2 times. Just once is enough. the active site will change. It will no longer be complementary. This is a very easy question. This is the description. Explanation, because this is the trypsin that is produced in the small intestine. And the small intestine, the enzymes there, they work best in acidic conditions. It is produced in the small intestine and by another organ. Which organ?
Pancreas.
Correct. And what provides the alkaline conditions? Like the hydrochloric acid
Right.
here provides, correct, perfect. So this also can be mentioned because it's out of 6. Don't forget it's out of 6. So you have hydrochloric acid here. It will give me the acidic conditions, and the bile contains sodium bicarbonate or an alkaline that will neutralize the environment. This is very good. Very easy question, by the way. And it's good it's out of 6 marks. So now you should take the 6 out of the 6. Maltase is a digestive enzyme that acts in the small intestine. State the exact location of maltase in the small intestine. Where exactly in the small intestine is the maltase produced and working? This is a tricky question, by the way.
Duodenum?
No, the
The
duodenum does not do
epithelium.
any type of digestion. Yes, the epithelium of the small intestine, the lining of the small intestine, the epithelial lining of the small intestine. And here you do not mention the pancreas because they're specifically talking about the small intestine. So it's the epithelial lining of the small intestine. The duodenum does not do any digestion. It's clearly mentioned in your notes. The duodenum only is a receiver. It receives bile and pancreatic juices. Okay, plants. Plants are very important. This is which device we took it that measures the distance of water here over time. Tell me the name of this device. Measure the amount of water absorbed by the plant over time. Give me a name. Omar, what's the name of this device that measures the amount of water absorbed over time?
I don't know.
Photo. It's called what? It has a name. Photo what?
Pottometer.
Yes, photometer. We took it.
Yeah.
You have 2 types of photometer, the bubble photometer and the mass photometer. And this is a 2-in-1 photometer, if you noticed. It measures the mass before and after, and it also measures the distance traveled by the water. So this is a 2-in-1 photometer. We change the environmental conditions the leafy shoot is exposed to, and we measure the amount of water and the change in mass. So this is a very smart photometer. What does it measure? State the name of the process, Omar, by which the leafy shoots lose water. How does the leaf loses water? Very easy question.
Transpiration.
Correct. Very easy. Transpiration. They always start with an easy question, and then before the shoot is inserted into the jar, it must be recut underwater. So when we cut it underwater. Why? Why is it cut underwater? Hmm? This is also in your notes. Why do we cut this underwater? We don't cut the leafy shoot outside and then we put it in the water, but we cut it underwater. Why? Why, Omar?
I don't know.
Uh, think. Let me choose someone. Jolly, why do we cut the plant underwater? So we
To
prevent
ensure
what?
continuous water,
Excuse me?
ensure that we have continuous water for the
No, no, to prevent the entrance of air bubbles into the plant. Again, this is also in your notes, in year 9 notes. You don't focus on every detail. We cut this plant underwater because if we cut it outside, air bubbles will enter into the xylem and phloem, and it will interfere with the transport of materials inside the plant. So we cut it underwater to prevent air bubbles. This is important. This is practical, and it can come also in paper 6. So I'll highlight that. Why it is cut underwater? To prevent air bubbles. I highlighted this for you. State the purpose of the oil layer on the top of the water. Now, this is easy. Why do we put oil? What's the reason why we always put oil?
To prevent the evaporation so it does not affect the volume of water.
Correct. Perfect. To prevent evaporation. Very nice and easy. Using the information, describe one method that can be used to determine how much water is lost from the leafy shoot. So, how can we know the amount of water lost from the shoot? Very easy.
Measure the mass loss.
Very good. Or measure the
Measure
what?
the water uptake.
Very good. But yes, just I want to be more accurate and say you are right. We will measure the amount of mass lost after 5 minutes. We will measure the amount of water uptake after 10 minutes. So always set a set time. Okay. So we will either measure mass loss, okay, or uptake of water over time. And this will help me, this will remind me that anytime you are measuring a gas or you are doing any measurement, even in paper 6, the question, the 6-mark investigation question, I will measure the volume of gas. Yes, don't just say I will measure the volume of gas forever. I'll measure the volume of gas over time. I'm going to measure the amount of water displaced over time, over a set amount of time. This is another graph. Temperature on the x-axis and the rate of water on the y-axis. Someone was asking me, can I start the graph from a number not 0? Yes, you can. But what's important is that the values should be equally distributed. See, 25, 27, 29. There's a value of 2 every 5 boxes. So this is important. Using the information in figure 3.2, calculate how much water would be lost from 1 meter squared of leaves in 12 hours if the plants were kept at 35 degrees, include the unit. So what will you do here? Again, using the information, calculate how much water is lost from 1 meter squared of leaves in 12 hours if the plants were kept at 35 degrees. Look at the y-axis. This is the rate of water lost in grams per meter squared per second. So what do you do? Where do you go on the x-axis? I don't want to answer it. You will answer it because I will not sit for the exam. You will. On the exam, what will you do? If the plants were kept at 35 degrees. So where is the first thing you will go to? You will go to which temperature? Huh?
35.
Yes, you will go to 35 degrees. And then you will read the rate of water loss at 35 degrees. I'll go to 35 degrees. I'll move up here, and then I'll move here. I have to figure out the value of this line. This is 0.20, and then this is 0.30, and I have 5 boxes in between. So what is the value of every small box?
0.2.
Correct. How did I get it? 5 divided by... How did I get the value of one small box? You are correct. I just want to show everyone how I got it. You said 0.2, 0.3. How did you get the value of every box? Tell me, how did you get it? It's correct. It is 0.2. It's 0.22, 0.24, 0.26, 0.28, 0.3. You are right. You're not telling me how you got it. So what's this value? What's this value? 0.2? Who gave me the answer? This is 0.28. So the rate of water loss is 0.28 grams per meter squared per second. Now they want it in 12 hours. What do you do? The answer is not 0.28. This is 0.28 per second. How do you convert it per hour? And then per 12 hours. I will not give you the answer. Someone give me the answer. Layla.
It
1
wasn't
second
me,
is how many hours? 1 second is how many minutes? Layla? Who is speaking now? Let me see.
ازاي miss 1 second how many minutes
One, صحيح. I mean, to convert second to minutes, what do you do?
times 60
Yes, شطورة. So 1 second is 60 minutes. And then I will convert the minutes
miss
to
you mean
hours. What do you
العكس
هي يا حبيبتي هي in seconds. احنا عايزين نقلبها what will how يعني in 1 second the rate will be 0.28 grams. It will absorb 0.28 grams in 1 second. How many grams of water will it absorb in 12 hours? You understand the question?
yes
This
but
plant
it's
will
supposed to
absorb
be 1 minute has 60 seconds
صح انت صح انت صح انا قصدي كده 1 minute is 60 seconds. Okay, 60 seconds. So
تمام
and then 1 hour is how many minutes?
60 minutes
Bro, 1 hour is 60 minutes. So if it will absorb 0.28 grams in 1 second, how many grams of water will it absorb in 12 hours?
0.28 times 3 اللي هو 60 times 60
Bravo, yes. Yes, who told you that this is wrong? 0.28 times 60. This is converting it into knowing how many grams it will absorb in 1 minute. And then we will multiply by 60 again. We will know how much water is absorbed in 1 hour. And then we want in 12 hours. So we will multiply by
12
12. Yes, this is right. Okay, let's look at the marks. I know you are now in doubt. I'll show you. This is not a nice question. This is number 3. I doubt that you will be able to do it alone on the exam. Where is 3? Is this the right mark scheme anyway? Yes. Look, 3BI. Look what did they do. This is the pen. Okay. Look, 0.28. This is what we got from the graph. Multiply by 60 to convert it to minutes. Multiplied by 60 to know how much water was absorbed in 1 hour. Multiplied by 12 to know how much water is absorbed in 12 hours. So yes, 0.28 times 12 to know how much water is absorbed in 1 minute times 60 times 60 again to know how much water is absorbed in 1 hour. But we want to know how much water is absorbed in 12 hours times 12. The answer will be this number, 12,000 or 12096 grams per meter squared. Will you know how to do this alone on the exam? I don't think so. Most of you will be able to get the number from the graph. So you will reach 0.28, but I don't think you will be able to get the correct number to get how much water is absorbed in 12 hours. So we multiplied 0.28 by 60 to know how much water was absorbed in 1 minute. And then we multiplied by 60 again to know how much water was absorbed in 1 hour. And then we multiplied by 12 to know how much water was absorbed in 12 hours. And you will get a number. This is the number. And you will write the whole number, okay? Because they did not tell you round it up to one number or 2 decimal places, and you will include the unit. So this is a difficult question. Here it is testing your mathematical skills, which this is not something that you are very strong at. And the unit will be gram per meter squared. Here, do you all know how we got this question now? I'm going to highlight it because this question, you always get a mathematical question on the exam, unfortunately, and we cannot help it. Now we all know what we did. Anyone has any questions? Using the information, describe and explain again the effect of increasing temperature on the rate of water loss in this species of plant. 5 marks. So how will you describe and explain? Can you describe first? Let's break it down bit by bit. Can you describe this graph?
Yes, we can
You
can
cannot
see
see
the
the graph?
screen.
This is strange. 1 minute. Can you now see the screen?
Yes.
Okay,
Yes.
before I move on, do you all understood this question, this mathematical question? Do you understand it now? Or anyone is confused? Okay, this question is not a nice question, so please go back and solve it alone so that you practice these kinds of questions. Now, here they want us to describe and explain the effect of increasing temperature on the rate of water loss in this species of plant. 5 marks. So we will describe and explain this graph. Who can describe it first? Hmm? The describing part is very important. Don't stay silent because whether you like it or you don't, you have an exam and you will have to answer. Yusuf Osama, can you describe this graph? Yusuf?
At 25 degrees Celsius, the rate of water loss
Okay.
from the leaves was
0.12, 0.14, 0.16, 0.16. It was 0.16 grams per meter squared per second. And then what?
and then as the temperature increases, the rate of water loss from the leaves increases. For
Very
example,
good.
at 35 degrees Celsius,
Mm,
the rate was 0.28.
perfect. And then it will increase faster or slower?
Then the rate of water loss increases faster until until at 45 degrees Celsius. It was the highest at 0.54.
246. Okay, perfect. I want to also show you something. This is an increase and this is an increase. But the difference between A and B is that B, the increase is faster. Okay, I think you already know that. So this is the description. Perfect. Can you explain why, as the temperature increases, the rate of water loss from the leaf increases?
Because particles gain energy, move faster by kinetic energy.
Excellent. As the temperature is increasing, the molecules will gain kinetic energy. Always mention kinetic energy with temperature. Okay. Why is this important? Or how will this affect the rate of water loss? This is not enough. It's the beginning of the answer. So the describe part is done. Now the explain part. You said as temperature increases, the molecules gain kinetic energy. You are correct. Number one, which molecules are we talking about? And what will this affect? Why the plant absorbs more water when it is hotter?
Increase the rate of transpiration.
Correct? Correct. It will increase rate of transpiration. What else will you say? What is transpiration? You will not define it?
Loss
5
of water
marks.
from
Of course, it's the loss of. First, let's define it properly, like we studied. We start with the word evaporation. Transpiration is the evaporation of what?
of water from the
Of
mesophyll leaves, mesophyll cells, sorry,
correct. It's the evaporation of
followed by
water, followed by
followed by
the
evaporation from
diffusion of water vapor from the stomata. There's a difference. So transpiration is the evaporation of water, only just water, because it's still a liquid. It's the evaporation of water from the surface of the mesophyll cells, correct? Followed by what? Diffusion of water vapor. Now it turned into a gas from the open stomata. This definition itself will give you 2 marks because you mentioned that we start by evaporation and it is followed by diffusion, and we mentioned these stomata from the open stomata. And this needs kinetic energy. This needs movement of water. First, you have here the guard cells. These are the guard cells. And you have here the mesophyll cells. Transpiration. Let's break down the definition. We did this 100 times. The water, let's draw it in blue. The water is here. First, the water will evaporate from the surface of the mesophyll cells. Evaporate means it will change from liquid to gas. So now it is gas droplets. These are the water vapor droplets. So this is the first part of the definition, that the water will evaporate from the surface of the mesophyll cells, and then it will diffuse out of the stomata as gas. And all this needs kinetic energy, because the kinetic energy will make the water molecules move faster. It will make the evaporation step very quick, and it will make diffusion of the molecules very quick, because of course you know that the higher the temperature, the more the kinetic energy and the faster the diffusion. So in total, the faster or the more the transpiration. So when you come to explain here, you write a lot of things. The increase in temperature will give the molecules kinetic energy. Which molecules are we talking about? We are specifically talking about water molecules, not any molecule, because this is what is being transpired out of the plant. Water will transpire faster. The rate of transpiration will be faster. This is another mark. And you will explain transpiration. Transpiration is a two-step procedure. Number one, I have the evaporation of water, just water, from the surface of the mesophyll cells, followed by step two, the diffusion of the water vapor, okay, from the opening through the stomach. So here you mentioned 4 points: kinetic energy, transpiration, evaporation, and diffusion, and water vapor. I think this is going to give you a mark. And you said open stomata. This is going to also give you the 5 out of the 5. Can you add something in case you are worried and you want to include everything? Why is it important that the plant loses water? Why does the plant transpire anyway at very high temperatures? Why does the plant lose water on a very hot day?
To prevent wilting.
You are correct. I keep on absorbing water from the roots, yes, to prevent wilting. Okay, another reason. Like us, why do we sweat on a very hot day?
For the temperature. يعني temperature بتاع البلانت.
Yes, you are right. So that the temperature of the plant does not rise very high, so that it cools down the plant. You are right. Okay, so these keywords, all of them, they have to be present. That's why it's out of 5 marks. Kinetic energy, transpiration, evaporation of water, diffusion, faster diffusion, because the faster or the more or the higher kinetic energy will lead to faster diffusion. Water vapor, it has to be there, water vapor. Water vapor, open stomata. All these are keywords. All these are keywords. That's why it's out of 5 marks. Let's look at the marks. Cooling down. We don't want to denature the enzymes. When it's out of 5 marks, if I am you, I will worry. So this is 3B. Where are we? Oh, we're right there. So temperature increases, rate of water loss increases. We said that. We said the numbers in the description. Any correct comparative data code, we said Yusuf described it very well. Water vapor is lost through the stomata. We said it. We can also mention guard cells, evaporation from the surface of the mesophyll layer into the air spaces. We said that. Diffusion, you even can define a diffusion down upon concentrated ingredient. Increase in temperature increases kinetic energy. We said it. Faster rate of diffusion. We said it. Stomata is open. You said it. Any valid point, any 5 from, so you don't have to mention all those 10 points, but we mentioned most of them, and it cools the plant. We even said that. Okay, that is why it's out of 5 marks, because there are so many keywords involved in this question. This question is easy, but I'm going to also highlight it because this is the type of question that you should practice before your exam. Okay, describe and explain. Suggest why the mass of the water in the apparatus does not change when the leaf is kept at 100% relative humidity. So when the humidity increases, why does the mass of the water in the apparatus does not change?
Because as humidity increases, transpiration rate decreases.
Correct, because the transpiration will decrease as humidity decreases. Okay, I just want another sentence to clarify why will the mass does not change. Will there be transpiration? No, you said it will decrease. But what will the mass of water in and out of the plant be for the mass to stay the same? So this means what? The amount of water in equals the amount of water.
Osmoregulation.
Osmoregulation, yes, but you are right. But we say this in humans. We don't say osmoregulation in plants. The plant absorbs water and it loses water. The amount of water coming inside the plant is equal to the amount of water
Lost.
Yes, I'm telling you that because it's 2 marks. If it was only 1 mark, just saying a decrease in transpiration is enough. But 2 marks, no. So you will say that the amount of water absorbed is equal to the amount of water lost. Okay, there is no change in water potential. The water potential inside the plant is equal to the water potential outside the plant. So there is no transpiration. So there is no diffusion or there is no evaporation of water. Okay. Look at my screen because I'm sure you're telling yourself that I'm asking you to write more than what is needed. No diffusion of water vapor, no water potential gradient described. So what we said is correct. In other words, the water potential inside equals the water potential outside. Water potential means water amount or water concentration. No change in the concentration of water. That's why the plant will not transpire and it will not lose weight. I hope it's clear. Again, I'm going to mark this question because I don't think you will write the word water vapor or water potential. Even at very low humidities, the leaf shoot does not wilt. Why does the leaf does not wilt? This is a very easy question. Why does the leaf does not wilt? Let's finish. When do plants wilt? When they have a
When
supply
water
of
loss
water
is
or
more
they
than
don't have
water
water? Excuse me?
when water loss is more than the water that they have. Yeah.
Correct. Now, why will they not wilt? Because there is water loss or because there is no water loss or they have water?
They have water.
Correct. They have water. Okay. The investigation on the effect of temperature was done at a relative humidity of 20%. The investigation was repeated at a relative humidity of 80, so the humidity increased, and all the other conditions were kept the same. So they only increased the humidity. Predict how the water loss will differ from the trend. Sketch your prediction on figure 3.2. So I'll go to figure 3.2. Of course, I messed it up, but you will not do this on the exam. What will you do to show how the rate of water is affected by increase in humidity? How will you sketch?
As the temperature increases هنخلي ال line steeper.
صح. Steeper, the rate will increase or decrease?
الريت هينكريس لما temperature يبقى أعلى.
They did not say that the temperature increased. They said that the humidity decreased. The humidity increased. See, the humidity
اوكي لو
increased from 2
humidity
to
يبقى
8.
هتdecrease هتبقى
Bravo.
less steep.
Yes, let's see. So how will you do it? Yani, how will you catch it? What you're saying is right. How will you represent this on the graph? You will draw a line above the line that's already present or below it.
below.
Yes. So you will draw a line exactly looking at the line that you have, but below it. Anywhere below it. Okay? Let's look at the next thing. Line draw below the original line. Any line just below it to show that there is a general decrease in the rate of water loss as the humidity increases. Okay, finish. Describe the structure of a DNA molecule. 4 marks, my goodness. What will you say here?
DNA carry genetic information. DNA is a double helix.
Yes, double helix. I'm talking about structure here. So double helix. What else? How many strands is it made up of?
2.
Correct? 2 strands. Okay. What else will you say about the structure? These 2 strands, what do they contain? What are they made up
Bases.
of? You studied. Yes, you are correct. They contain bases.
HCG.
Which bases?
AT.
Bravo. Huh? CG.
CNG.
Correct. C and G. And this is called complementary base
Complementary
pairing.
base pairing.
Correct. And how are these bases bonded together? What are the bonds that hold the bases?
Hydrogen.
Correct. Hydrogen bond. Write everything you know about the structure of the DNA. And you did study that the DNA is a polymer. It's a large molecule. If you don't remember polymer, it's okay. Made up of smaller units. What are these small units called?
Monomers.
Yes. What is the monomer that makes up the DNA? We
Nucleotide.
studied it. Shakura, nucleotide, made up of the sugar and the phosphate and the base. And you have another nucleotide opposing it. If you know how to draw it, good for you. If you don't, you can simply draw a double helix. Okay? So this is everything about the structure of the DNA. Outline how antibiotic resistance develops in a population of bacteria. Now, this question is highly repeated. You should memorize it by now. What do you start with? I want to finish this exam.
That non-resistant bacteria will die while resistant bacteria will survive.
Okay, but you jumped. There, we start with something. There's something that must be present before mentioning what you said. So here you started from the middle. What happens so that I have 2 types of bacteria? I have resistant and non-resistant. First, I have what?
Mutation.
Someone remind her. Yes, correct. First, I have mutation. And this mutation will cause what?
Variation.
Variation.
Correct. Variation. And you can define mutation if you want. And because I have variation, I will have 2 types of bacteria, at least 2. One that is resistant and one that is non-resistant. And then which one will survive and which one will not survive?
The resistance will survive.
Of course, the resistant will survive and the non-resistant will die. And when they survive and the others die, this will increase or decrease competition.
Will decrease competition.
Decrease.
Correct, it will decrease competition. Now, the resistant bacteria, what will be their fate? What will happen to them, huh?
They will
When they're happier.
reproduce.
Very good, they will reproduce. And when they reproduce, what happens?
Pass on their alleles.
Perfect. Pass on their favorable characteristics or their alleles. And with time, what will happen?
All the bacteria will be resistant.
Very good. All the bacteria now will be resistant. And this is an example of what? You have to end it by saying this is an example of when organisms change with time, because the nature will choose which is
Natural
more fit.
selection.
This is an example of evolution by natural selection. The word natural selection is a keyword. It has to be in the answer. It has to be in the answer. Look at the mark scheme. I'm sure that they tell you always in this type of question, natural selection has to be there. The word natural selection. And if this question was out of more marks, you will say that the antibiotic was the selective pressure and the favorable characteristics was the survival advantage. But you have to say natural selection. Suggest why scientists use differences in base sequences to classify the strains of the MRSA rather than using other methods. Why base sequences is used in classification? Again, we took it in classification. This is year 9. Why do we use base sequences in classification to classify organisms of different species? Give me
See
an
the
answer.
similarities, how similar, and the more
Yes,
similar, the more like related they are.
very good. And the more the base sequences are different, the more they are. Huh? So when the
Not
base sequences
related.
are similar, they are, yes, you are right. The more the base sequences are similar, the same species. And the more the base sequences are different, the more they are different. They're not the same species, not same species. We study different methods of classification, either anatomy or the external features, and the most accurate is the base sequences between organisms. This is the most accurate method. And everyone is unique. We have different base sequences, but the closer they are, the more we are of the same species. And the more different they are, the more they are of different species. This is classification. Explain why scientists are concerned that some strains of bacteria, such as S. aureus, have been resistant to antibiotics. Why is this a problem? Antibiotic resistance. This is the lesson drugs. Why is antibiotic resistance causing a concern to a lot of doctors? Can't imagine that you're not giving me an answer to such an easy question. Give me an answer. Omar,
We don't take the proper dose or the full dose.
your answer is not answering the question. I did not ask you, how do we develop resistance? I am asking you, why is resistance to antibiotics so bad?
They make bacteria harder to get rid of and illnesses
Yes,
harder
you
to
are
treat.
correct. Because the antibiotic will be ineffective, because I will not be able to treat infections. So the antibiotic is ineffective, and the infections, I cannot treat it. I can die from infections. I cannot treat it. Correct. Okay. Describe how the use of antibiotics can be managed to reduce the development of resistant strains of bacteria. Now, whoever answered me and told me because I'm taking it because when I don't need it, this is the answer. Describe how the use of antibiotics can be managed. So, what can I do to prevent antibiotic resistance? This is out of 2, so give me 3 ways I can prevent antibiotic resistance. Do I take it when I have a viral infection?
No.
No. Do I take it when I have headache? Do I take it when not needed? Okay. I only take it when needed. I take the correct dose. I only take it when it is prescribed by the doctor. I isolate the patients that are infected so that I do not spread the disease. I improve my hygiene when there is infection. That's why doctors, they wash their hands very well after they deal with patients so that they do not infect other patients. Suggest why this MRSA, this bacteria, is unlikely to be transmitted from a mother to her baby. Suggest
Because
question.
it's a bacterial infection.
No, but some bacterial infections can be transmitted from the mother to the baby. But why would this one precisely not be transmitted from the mother to the baby? What will prevent it from being transmitted? What
The
is the organ?
placenta.
Shatur. This is the organ that provides some protection, not 100% protection, but some protection. State the names of 3 natural processes involving bacteria that are important to ecosystems.
Fermentation,
Correct? Fermentation.
nitrification,
Shatur, nitrification. Nitrifying bacteria. Good. What else?
deamination,
Deamination. Yes, deamination. Okay. Uh, Also, give me other examples. Yes, you are right. Nitrogen fixing bacteria. Nitrogen fixing bacteria is fixing the bacteria from the air. I'll take it from the air, the nitrogen, and I'll convert it to nitrates. Deamination is removal of the nitrogen containing part. Each one is different. Nitrifying bacteria, in case you forgot, this is converting the ammonium compounds into nitrates. So each one of them is different. Nitrifying bacteria will convert the ammonium compounds into nitrates. Deamination is removal of the nitrogen containing part to create ammonium compound. Nitrogen fixing bacteria will fix the nitrogen from the air, convert it to nitrates. Fermentation, this is the anaerobic respiration in yeast. Very good answers. This is a nice question. Let me highlight it. Okay, it doesn't finish. This exam doesn't finish. State one other natural process that releases carbon dioxide into the atmosphere. Ecology. What releases carbon dioxide into the atmosphere?
Respiration.
Other than fires. Correct.
Respiration.
Respiration. Combustion. Yes, decomposition. Very good. State the name of one other greenhouse gas other than carbon dioxide.
Methane.
Yes, very good. Okay. Fine. Now this is the fire, estimated total number of fires, estimated average, I don't know what's this. Using the information in table 5.1, compare the data for the 2 managed locations with the data for the 3 natural locations. So this question, you will only break down the data. It's got nothing to do with how much you studied. You will go to the data, to the table, and you will compare. You will take numbers and compare one by one. For the 2 managed locations with the data for the 3 natural locations. So I'll go to the table. I have something called managed locations and what? Natural locations. Where are the 3 natural? Estimated average expansion rate of fires. The occurrence of fires globally using a 14-year period, and they tracked all the fires that were larger, therefore visible from space. Table 5.1 summarizes some of their data, categorizing the fires by location. The locations include natural ecosystems and land that is managed by people. And the expansion rate is the speed at which each fire becomes larger. So we will compare natural and natural and natural. So you will compare those 3 with those 2. This is the natural locations, and these are the managed locations. Managed means managed by people. You will compare, you will break down the numbers. You have estimated total number of fires. Of course, I can see that the number of fires is the highest in the grassland, and the number of fires are generally more or less, they're nearly the same. Like, you will compare the numbers, the managed agriculture land, and it is here managed land being deforested. The estimated average expansion rate, it is generally less, and it is generally more in the natural. Estimated average duration of the fire, it is generally also more in the natural and less in the managed. Of course, you will write the general conclusion and you will give numbers. Describe how the data in table 5.1 could be used to estimate the total area that was burnt during the 14-year period for each location. Now, this is not a nice question. How can you estimate the total area that was burnt during the 14-year period for each location? How can you do that? Just estimate, how can estimate? Look at this question. We want to know if, for example, you have the natural forest, the estimated total number of fires is this total number. The estimated average expansion rate of the fire per day is in 1 day. The estimated average duration of the fire is this per day. So how can you know if you know the amount of fire in 1 day? How can you know the amount of fire in a certain number, more than 1 day? What will you do? What will you do? How will you answer this question? Describe how the data could be used to estimate, to give you an estimate of the total area that was burnt during the 14-year period for each location. They did this over 14 years. How can you estimate how much is the total area that has burnt over the 14-year period? Give me an answer. You're all silent. You are on the exam. What will you do? What will you do? I'm silent. What's the easiest way to estimate? How will you know the amount of fires over a 14-year period? Who will give me an answer, or you will keep it empty? Layla.
Yes.
Okay, I know this question is a little bit tricky, but how can you, if they are calculating this data over a 14-year period and they estimated the average expansion rate and fires in 1 day, how can they estimate how much fires will happen over a 14-year period?
So
Total number.
these are in days.
This is
Oh,
the total number of fires over 14 years, and then the average number of the fire in 1 day, and the average duration in 1 day. So this is the total 14 years. And this is the average in 1 day, and this is the duration in 1 day. It's easy, by the way.
Cross multiplication.
No, we will not cross multiply. You know what we will do? Simply, you will multiply the total number of fires by the average of the fires in 1 day by the duration of the fire in 1 day. So you will multiply the whole row for every one of them, and you will estimate how many fires over 14 years, the total area that was burnt over the 14 years. Now, let me just share the marking in case I'm making up math, because I am horrible in math, I admit. This is the only obviously what will answer this is 5B. Okay, I guess I am right this time. I will multiply the number of the fires by the expansion rate by the duration for each row. This is the only way that you can get it. Now, I'm going to highlight this question because they're very unfair. We are here studying biology, not math, but they get you mathematical questions, and these mathematical questions we are all not good at, including me. I will not tell you that I'm better. So just practice them. There's nothing to do about it. Describe the possible consequences of destruction, of deforestation. What are the consequences of deforestation?
loss of habitat, increase of CO2 in the atmosphere.
Very good. Loss of habitat. I want at least 3. More CO2, correct. And more CO2 is going to result in what?
Global warming,
Very
or
global
climate
warming.
change.
Yes, excellent. What else? Decrease in
In oxygen level.
Biodiversity.
Yes, decrease in biodiversity. Very good. How will this affect food chains? Less food, less shelters, less plants, less animal, loss of genetic pool. Okay, I just want to finish. Anyone has any questions? I don't think this is an easy question. Okay, this is the human gas exchange system. God, this exam is so long. Where is what affects where is the hairs in the nose? No, letter A. What is the function? The hairs in the nose. What is the function of the hairs in the nose?
Mechanical barrier.
Excellent. Mechanical barrier. What does it prevent or trap?
Trap dust particles.
Yes, dust. Good. Prevents collapse of the airway. Which letter here prevents collapse of? Yes, correct. Cartilage. J. Okay. And then contracts to decrease the pressure in the thorax. What will contract to decrease the pressure in the thorax? The diaphragm is already through use, so we will not use the diaphragm. What else contracts with the diaphragm?
The intercostal muscles.
Yes, very good. The external or the internal?
External.
Correct. The external intercostal muscles. Okay, very good. Now the diaphragm, where is it and what is the function of the diaphragm? Mm. What is the function of the diaphragm? The function of the diaphragm, we can copy this function, by the way. It will contract during inhalation to decrease the pressure in the thorax and to increase the volume. Where is the diaphragm here in this picture? Letter. Louis, which letter here is the diaphragm?
C.
That's correct. C. I want to finish this exam. What protects the lungs from mechanical damage? This is so easy.
Rib cage.
Yes, the rib cage. What is the letter that you will choose?
E.
Correct. This is very, by the way, tricky. E is the rib cage. Again, this is year 9 notes. F are the external intercostal muscles, and D are the internal intercostal muscles. So correct, it is E. So here, protect the lungs. Here, it is E, rib cage. Good girl. Site of gas exchange. Let's go quickly. What is the site of the gas exchange? This is very easy.
Lungs.
Lungs.
Alveoli.
Yes, yes, shatura. H. Good. Describe and explain how the alveoli are adapted for gas exchange. Again, describe and explain. Now, you took a list of adaptations for the alveoli. We want 3 adaptations. Who will remember?
Thin.
Yes, they're thin. Thin for
Large
faster what?
surface area.
Yes,
For
you are
faster
correct.
diffusion.
Correct.
Many
Large surface
capillaries.
area. Correct. Many capillaries to maintain a concentration gradient. Also, it's moist so that what is called the gases can dissolve. It's porous, it's surrounded by a lot of capillaries and a rare finish. Explain the differences in composition between inspired and expired air. Now we took a table. Let's talk about the concentrations of oxygen and carbon dioxide and the water vapor and the temperature. In inspired air, how much oxygen in?
21.
21%.
Yes, very good. 20 to 21%. How much out?
16%.
16%, so less. CO2, how much in?
0.04.
Very good. How much out?
4%.
4%, so more out. Water vapor. We don't have numbers here, but what more outside? Yes, saturated with water vapor. The temperature, it's warmer in or out? Always the air coming out of your mouth is warmer. State where this change is detected and how the body responds to this change. What is the question? Physical activity changes the concentration of carbon dioxide in the body. This is easy. How is it detected and what is the response? High carbon dioxide is detected by the
By the brain.
Yes, by the brain. Now, 1 minute. This question here, explain the differences. We need to explain. So there's an explanation here. So explain not only the comparative differences we've explained. Oxygen outside is less because I need the oxygen in aerobic respiration. Carbon dioxide is more when we exhale because it's a waste product of respiration. Water vapor evaporates. There is an explanation in the table in your notes, so you write the explanation. Yes, it is detected by the brain. High carbon dioxide levels are detected by the brain. How does the brain respond?
Increasing the depth rate.
Correct. If we are going to discuss it in terms of coordination, the brain will send a motor impulse, if you want a perfect answer, to your muscles in your lungs so that they contract. Which muscles we're talking about that the contract? The diaphragm, and the external intercostal muscles. So they contract and they start to increase the rate, like you said, and depth of what? Huh? Correct. And why do we breathe faster when there are high CO2 levels?
To decrease the oxygen depth.
Very good. To decrease the oxygen depth and to return the oxygen levels back to normal, or the CO2 levels back to normal. And this is an example of what? Example of? Very good. By negative feedback. Now, why did I give you this full answer? Because that same question can be out of 4 marks, not 2 marks. That same question, and it came in another exam. It was out of 4 marks, so they want all the keywords. So I would highlight it. It's an easy question, but it can be out of 2 or 4. Say the name of a solution that can be used to test for the presence of carbon dioxide.
Lime water.
Yes, lime water. Okay, this exam doesn't finish? Okay, it's finished, finally. Oh my goodness. Now, this exam, in my opinion, was a difficult exam. Difficult exams, there is an advantage and a disadvantage. That the curve is low, but you come out of the exam feeling very depressed. It really discourages students and they don't study for the remaining papers. But the curve is low. Now, this exam had a lot of questions from year 9. So if you don't yet study year 9, you will suffer in an exam like this. So again, I'm telling you, if you took the core last year, you can study it lightly. But if you did not take the core last year, you will study it as intensely as you would year 10 notes. I'm going to send you this exam also because there are a lot of highlighted questions. There are 2 graphs that you wanted to describe and explain. So graphs are very important. There are 2 mathematical equations that were not easy. This one was one mathematical equation, and the other mathematical equation here, this one, that was also not easy. It was not nice. The problem with mathematical equations is that we cannot study them. We just practice them because any mathematical equation can pop up. And I advise you to always solve variant 3 in paper 4 because variant 3 is the hardest variant. This was variant 1. Oh my goodness, this is variant 1 and it was not easy. Okay. I'll meet you on Tuesday, and on Tuesday we'll solve a paper 6. We solved a lot of paper 4. I think on Tuesday we'll solve paper 6. If you have any questions, send me. Fine? Any questions before I go? Okay, bye.